Oh, very good evening, my dear friends. Today's our eminent resource person, Dr. B.RUKMINI Devi Madam Garu, Assistant Professor, Department of Botany, GDC NARSAPU, Medak District. Madam is going to explain the general characteristic features of Mastigomycotina. Madam, please share your screen and start your session.
Okay, sir. Now we will discuss about hypocytidiomycetes characteristic features. These hypocytidiomycetes members are aquatic freshwater forms and marine fungi. Here, these members will produce zoospores during reproduction. These zoospores are anteriorly uniflagellated zoospores. Thallus is holocarpic thallus or eucarpic. This eucarpic is again classified into monocentric or polycentric. Monocentric means a single reproductive structure is produced apart from vegetative thallus. Polycentric means more than one reproductive structure is produced apart from this vegetative structure. That kind of fungi you will see in this hypocytidiomycetes. Vegetative system is rhizoidal or hyphae-like structure. If it is hyphae-like structure means that consists intracalary swellings. In between some swellings you will find in this hypha. That kind of hyphae-like structure you will find in this vegetative plant body, or this vegetative plant body may be in the form of rhizoidal kind of structures. Cell walls contains chitin or cellulose. Sometimes in addition to chitin, cellulose also present in these members. Sporangia are inoperculate type of sporangia. Zoospores are either completely differentiated in the sporangium. Zoospore differentiate outside the sporangium. That condition also you will find in some of the hypocytidiomycetes members. Here, these are morphologically similar to chytidiomycetes, but it differs from them in the structure and anterior flagellation of the zoospores. In some features, these are similar to chytidiomycetes, but these are differs from chytidiomycetes in having this anterior flagellation of zoospores. Now we will discuss about classification of hypocytidials. Based on the organization, morphology and development of the thallus, here these are classified into three families. One is Anasolpidiaceae, second one is Rhizoidiomycetaceae, third one is Hypocytriaceae. Now we will discuss about phylogeny of hypocytidiomycetes. On the basis of differences in the molecular weights of rRNA, Lovett and Hasselby found that anisocytrids could not be placed in any other major class of fungi. According to Batnicki and Garcia, they are also placed in anisocytrids outside the main phylogenetic line of fungi. These are having so many similarities with the oomycetes. According to Leeson, on the basis of regulation of glutamic dehydrogenase activity, they proposed that anisocytrids are precursors of both the chytidiomycetes and oomycetes. The hypocytidiomycetes differ from the chytidiomycetes on the basis of pathways of lysine synthesis, the nature of flagella as well as the cell wall composition. On this characters basis, these are differs from chytidiomycetes. One is a lysine synthesis pathway and another one is a composition of cell wall. These are differs from chytidiomycetes. In hypocytidiomycetes, a diaminopimelic pathway of lysine synthesis occurs, but in chytidiomycetes, aminoadipic synthesis occurs. Also, difference in the position of the flagellum on the planospores. That kind of difference also you will find between this hypocytidiomycetes and chytidiomycetes. Now coming to oomycetes. Vegetative body is filamentous and cenocytic except the unicellular lesnidiase. Holocarpic or eucarpic, this oomycetes members. That means Some members are holocarpic in nature. That means this unicellular structure entirely modifies into reproductive structure on maturation. That kind of fungi are called as holocarpic fungi. That kind of holocarpic members also you will find in this oomycetes. Next, eucarpic fungi also you will see in this oomycetes members. Eucarpic means some portion of this particular vegetative body modifies into reproductive structure. Remaining portion functions as a vegetative structure. That kind of members are called eucarpic fungi. So here in this oomycetes members you will see holocarpic and eucarpic. Cell wall contains cellulose and glucans. Here chitin is absent. Two types of zoospores formation you will see in the life cycle of oomycetes members. One is primary zoospore, second one is secondary zoospore. Primary zoospore is the first formed zoospore. Here flagella are located anteriorly. Here these are released from the zoosporangia. These will be germinated and produce as a secondary zoospores. Next, coming to secondary zoospores. Secondary zoospore is a reniform. That means a kidney shaped structure. This is also called as a bean shaped structure. Here flagellum is located at the lateral side. That is why the secondary zoospores are called as laterally flagellated zoospores. Zoosporangium and zoospores are the major dispersal agents for most of the species. Coming to sexual reproduction. Here sexual reproduction is heterogamous type of sexual reproduction. That means here two types of gametes formation you will see. One is female gamete and another one is male gamete. Female gamete is called as oogonia. Male gamete is called as antheridia. Female gamete is produced by oogonium. This is also called as oosphere. Depending on the taxa, there may be one to many oospheres for oogonium. Here female gametes also called as oospheres. These are produced inside the oogonium. Depends on taxa, the number of oospheres or female gametes formation differs. In one single oogonium, sometimes only single female gamete or oosphere formation will take place. Sometimes in single oogonium, more oospheres or female gametes formation will take place. Male gamete is produced inside the antheridium and that is transferred to oogonium by gametangial contact. So, by gametangial contact, this male gamete reaches female gamete, that is oogonium. So, whenever it reaches this oogonium, then what will happen? Male nuclei is released from this antheridia that will be migrated towards the oosphere. So, here two types of fertilization you will see. One is homothallic, next one is heterothallic. Homothallic means self-fertile. Heterothallic means opposite mating types are required for sexual reproduction. So that kind of sexual reproduction you will find in this oomycetes members. One is a homothallic type of sexual reproduction and another one is a heterothallic type of sexual reproduction. Here swimming sperms are absent in this oomycetes members. Here this sexual reproduction process is also called as gametangial copulation. Here male and female gametes both will undergo meiosis. So eggs and sperms are products of meiosis. So these are haploid in nature. This male gamete as well as female gametes both are products of meiosis. That is why these are haploid in nature. As a result of fusion of this male and female gametes, zygote will be formed, which is diploid in nature. This diploid zygote is surrounded by one thick ball, which is having high resistance. So this thick walled resistant oospore, which is undergoes germination process. Okay, after formation of thick resistant wall around this zygote, then that is called as a oospore. This will undergo germination process. As a result of germination of oospore, that will be gives rise to vegetative hyphae. That hyphae is diploid in condition. So this will reproduce asexually by the production of zoospores. As a result of sexual reproduction, one diploid zygote will form. This diploid zygote germinates and gives rise to vegetative hyphae that is also diploid in condition. This diploid vegetative hyphae reproduced by asexual method. So as a result of this asexual reproduction of vegetative hyphae, zoospore formation will take place. This is the life cycle of oomycetes members. How this sexual reproduction and asexual reproduction will take place here explained in this flow chart. In this oomycetes, 4 orders are there. One is lesnidiales, next one is leptometales, third one is saprolegniales, fourth one is peronosporales. Peronosporales order is most well known for pathogens. These are causes many diseases to crop plants. This peronosporales order divided into 3 families. One is Pithiaceae, next one is Peronosporaceae, third one is Albuzinaceae. Pithiaceae consists of 2 important genera. One is called as Pythium and Phytophthora. Peronosporaceae order is comprises 1 genera that is called Plasmopora. Next, third one is Albuzinaceae. This Albuzinaceae consists of 1 genus that is called as Albugo. 3 families are present in this Peronosporales order. One is Pithiaceae, second one is Peronosporaceae, third one is Albuzinaceae. Albuzinaceae comprises single genus that is Albugo. Peronosporaceae also comprises 1 genus that is Plasmopora. Pithiaceae comprises 2 genera. The most economically important group of oomycetes is Peronosporales. It consists of late blight of potato fungus. In potatoes, late blight disease is caused by 1 fungus. That fungus name is Phytophthora infestans. This Phytophthora infestans causes late blight disease in potato. Some other genera also causes diseases. Those members are Peronospora, Bremia, Plasmopora. These will cause downy mildew diseases, damping of disease. Pythium species, these members causes these diseases. And white rust disease is caused by Albugo. These are the diseases caused by this oomycetes members. With this, I am concluding this session. Thank you, sir, for giving me this opportunity.
Yes, madam. You in an excellent manner, madam. The clear cut.
Sorry for the inconvenience, sir. So many times I got disconnected due to some technical issues.
No, madam. The issues due to internet issues, we know that. So really an excellent class, madam. Thank you very much. So tomorrow also definitely we will have the another session tomorrow. So my dear friends, utilize this wonderful opportunity. So madam has huge experience regarding of this area. So madam, thank you very much. So tomorrow we will meet again. Okay. Thank you, madam.
Thank you, sir. Thank you, sir.
Thank you, madam.